Great. So thank you all for joining us today, especially those of you who have had to travel far to get here. Before we get started, I do have a few housekeeping items that I want to share with everyone. So first, please silence your cell phones and your laptops so we don't hear chimes throughout the day. There are also charging stations in the perimeter of the room if you need to charge cell phones or laptops or anything of that nature. This conference is being live streamed, so I just want everyone to be aware that there is a photographer and videographer in the room taking pictures and filming throughout the day. The restrooms are located out these side doors down the hallway. You'll see signage for that if you do need to use the restroom. You can also find additional restrooms at the bottom of the escalators right outside this back side of the room. The Wi-Fi code, if you would need it, is RDCA_DAP, and there's more information on each of the tables for login. And then also we're asking that you please follow us on Twitter. So if you're an active Twitter user, please make sure you're using our hashtags that we've identified. Those are also given on the tables in the inserts. And then if you have any questions throughout the event, you can either come to the c-path or NORD information tables or the registration table right outside the room. I also want to inform you that anyone who has a white lanyard is also a staff member, and we can answer any questions you may have. And then those of you who are watching us on live stream, you can email your questions to RDCA-DAP@c-path.org. And if your questions are not addressed today, we will follow up via email. And now I would like to introduce Dr. Joseph Sheeran, our CEO and President of Critical Path Institute. He joined us in April.
Thank you, Lindsay. Good morning. We from the Critical Path Institute are pleased and thrilled to launch together with NORD, the National Organization for Rare Disorders, and the FDA, the Food and Drug Administration, the Rare Disease Cures Accelerator Data and Analytics Platform, which we will call briefly RDCA-DAP. And I think that in our organization it will probably be called the DAP Initiative. The RDCA-DAP is part of a larger effort to accelerate progress in finding treatments and cures for patients with rare diseases. Another program in the RDCA Clinical Outcome Assessment, will create a consortium to develop patient-reported outcome tools. This has also been initiated by the same partners, c-path, NORD, and FDA. There are many rare diseases. It seems more than 7,000, and more than 17% is brain-related. The database analytics platform you're creating will enable us to obtain new insight in these rare diseases, not only within specific diseases, but also, we hope, across related diseases. c-path has successfully built databases over more than 8 years, and we have now data from more than 80,000 patients in those databases. The data consist of high-quality clinical data, which have permitted us to validate and obtain qualification or other regulatory endorsement for new drug development tools. The work we do is only possible when done in close collaboration with the stakeholders, including the regulators, industry, patient foundations, and academia. Building these databases needs the support and collaboration of all involved. We need access to clinical data from the industry, patient groups, and academia to succeed. Even more so, the RDCA-DAP will need to incorporate data from many sources. In addition to data from clinical trials conducted by industry and academia, we will want to access data from patients, hospitals, and any organization that can provide data within the frame of data privacy considerations. The data will be diverse in type and include not only numerical data, but also images, videos, genomic data, etc. The database will be able to handle these diverse data sets. Our quantitative medicine group is preparing to analyze these data sets with the most sophisticated tools available. We do not start empty-handed. Our partner, Nord, already has a registry based on patient-reported data, the IMCare database. c-path is already active in Duchenne disease, polycystic kidney disease, and Friedreich ataxia, for example. We want to include all these data in the RDCA-DAP. The database framework has been set up and is ready to accept data. So all of you who have data to contribute, please do not hesitate to contact us. This will be a project for the rare disease community as a whole, and we want and need your engagement every step of the way. We have created a rich program today, and you will hear more detail from the experts on what we plan to accomplish. So in summary, we are thrilled to be part of this new initiative, RDCA-DAP, and are looking forward to helping to find new treatments and cures for patients in need. We thank the FDA for providing the grant, making this initiative possible, and Nord for embarking with us on this major joint endeavor. Now I would like to introduce the President and CEO of our partner Nord, Peter Saltonstall. Peter, the floor is yours.
Thank you, Joseph. I'd like to add my thank you and welcome to everyone who's here today. And those of you that are online watching us, welcome as well. Nord is thrilled to be part of this innovative and patient-focused accelerator and data analytics program. And it really seems only appropriate to me that Nord, after 36 years of innovation, whether it be discovering and birthing the Orphan Drug Act, if you will, back in 1983, or building innovative programs for patient assistance back in 1987, when there was a need for patients to get access to their therapies. Or whether it be today in 2019, when we have more than 1,500,000 people a month coming to us on our website, looking for help, guidance, advice, any kind of information that can help them find a therapy. The key today is to remember that this accelerator is going to help us solve the problem of the fact that 95% of most rare disease patients today don't have an FDA-approved therapy. But I really want to thank the FDA, Janet Woodcock, her team, and the leadership that they've shown of recognizing the value of patient-reported and family-reported data and the value that that plays in the drug development process moving forward. And I just can't thank her enough for helping us make this all happen today. It prompts me to remember a meeting I had with her about 5 years ago when I sat with her and said, Janet, if there's anything that Nord could do to help expedite and de-risk the drug approval process, what would that be? And she looked at me and she said, longitudinal natural history data. And from there, today is the culmination, as Joseph said, the IMRARE data analytics, or the IMRARE registry program is a critical part of this new Cures Accelerator and data analytics platform. And so we welcome the opportunity to work with all of you. As Joseph said, we need data. We're accepting it immediately. Look forward to working with all of you today. If you have any questions about any aspect of the program, please speak to any of us that have the white lanyards on. And again, a special thanks to our partners and team, and a special thanks to the FDA for having the vision to recognize that we keep the patient at the center of this. It's going to be really successful. So thank you. Janet?
Well, good morning, everyone. I'm really delighted to be here, and as Peter and Joseph said, we're seeing a culmination of cumulative efforts we've put together over the years. Now they're coming together, and we're going to build something I think that'll be really important. So the Rare Disease Cures Accelerator Data and Analytics platform, like, why do we need this, and what is this about? Well, people with rare diseases tell us every day they need new therapies now. And of course, we know that, absent drug development programs, that's not going to happen possibly for many years. And so we need to get entrance into this space and make that pathway clear. But there are a lot of things that need to be done in order to develop a therapeutic for a rare disease. Most rare diseases don't have, as Peter said, any specific therapy for their disease. But last year, about half of CDER's new molecular entity approvals were actually for rare diseases. And this is really a big change, and I think it's room for optimism, and it shows kind of the concerted efforts that have been going on for a number of years. But we know there are thousands of other disorders and diseases that have no treatment right now. I think foundations and patient advocacy groups and others that have been trying to help in this space have realized that simply funding basic research, although it's necessary, it's really important, it's not enough to get those therapies into the hands of doctors and patients. You have to enable translation of research for that disease. So we have the basic science knowledge, but somehow we have to get it translated into the clinic. And the science that actually applies to any given rare disease and may lead to a cure actually may not come from the specific research that has been funded in that disease. It may come from left field, from cancer research or from this research or that research. But whatever it comes from, we need to grab it and enable it to be translated. And a lot of other activities are needed in order to bring that translation about. Sometimes, to start at the beginning, sometimes it's as basic as the disease definition and what are the characteristics of the disease. We may need, and I have seen, and it's been very helpful, consensus conferences, just to get agreement on what the disease is, what constitutes the disease, right? Many of these rare diseases, they're all different definitions, and people have different ideas of what they are. And data sharing is critical to get this right, to get the correct diagnostic criteria, because if you have one definition in this country and this definition over here, and the neurologists call it one thing and the ophthalmologists call it something else, and so forth, and pretty soon you don't have anything that you can work with, right? So part of this data sharing in this data platform is get all that information in one place so that people can look at it, and maybe a consensus effort might be needed on what are the diagnostic criteria for that disease. Seems like a simple thing, but for these rare diseases, it often isn't done. And when it has been done, it has led to progress. The second thing, once you have a disease in hand, in other words, not just a name of some famous scientist that's been put on the disease, but actually a set of diagnostic criteria that people can agree with, you need to understand the variability in that disease. Okay, so you can say, okay, so this person has this disease, but how do they present? How do they present, and what is the course? We know that for people with rare diseases, diagnosis can be delayed for up to a decade, right? They could wander around in the wilderness of specialists and never get a firm diagnosis, right? Because unfortunately, the presentation of the disease can be variable. And frankly, variability is one of the killers in drug development. So we have to understand variability. And then what is the course of the disease? Often, we know about the disease course from single interested investigators in different places who've published case series. And they have a very narrow view may depend on what specialty they are and what kind of people they see with that disease, or whatever. So data sharing, and you're going to hear this throughout my talk, data sharing is critical. We have to have all this information in the same place so we can at least know everything that somebody knows about this disease, okay? How can it present? How does it progress over time? What are the main symptoms you should watch? What is the variability? Some rare disorders are highly monotonic and they don't have a lot of variability. Most of them are quite variable. And then, of course, people now, since 2000, they've been looking at, and even earlier, really, they've been looking at the genetics of disease and genetic influences on the presentation. And this is critical to put all information together for the rare disease, along with the phenotypes, along with the disease course, along with the diagnostic criteria. So all of a sudden, you have something you can really work with. Then, of course, much of a research effort in a rare disease and in seeking a treatment goes to then looking at, okay, we know the natural history, we have defined the disease, and we kind of know the course, and we understand the variability. What's causing the disease? And it goes everything from what pathways are perturbed in the person, what is abnormal, how variable is that across patients? And really, sort of one of the real killers is, could this be a cluster of mechanistically distinct diseases that present the same way? And they're calling this disease Y, and yet it's caused by A or B or C, and A and B and C might need totally different interventions. And if you mix them all together in a trial, you're doomed, right? So, again, data sharing, critical to try and figure this all out, because just small samples, 1 or 2 people here and there, we're never going to figure out this really critical point. For disease intervention, finding treatments or cures, understanding mechanistically what's going on, what's causing the problem is key. And if it's several different mechanistically distinct problems, that has to be sorted out. Okay, so then you kind of understand what the disease is, you kind of understand its variability in the clinical course, you have an idea about the pathogenesis, and you want to then have an intervention, and you want to test it in people, right? And that's what we're looking for here, that's the cures, right? But then all of a sudden, there are things you don't know. Right? What measurable clinical outcomes actually occur in this disease? And are there standardized ways to measure them? Or you have to make up everything. You have to make up ways to measure these problems. Now, in another part of the forest, we're going to work on core outcome measures and so forth. But how quickly do these measures manifest themselves and change? How long would it take to measure a change in one of these measures? And then, of course, critically important, and people are already working on this, and these the NORD registry will help a lot, I think, is what matters to patients? What is the burden of disease from their point of view? And what can you measure that is really burdensome to patients about this disease? And what is the time course of change of that measurable outcome? So, and we also probably need to ask the patients what constitutes meaningful change. What would be for them? Is it, if it's stabilization, how much stabilization of disease? If it's amelioration of a symptom, how much is important to that patient? And okay, so then now you're pretty far down the pathway, right? But the only reason you've gotten there is you've had all this data that you've collected. You've been able to put it together, and you have a very strong picture in your mind of the disease, its pathogenesis, its natural history, the influence of different factors. What if you have a biologically plausible agent, okay, in hand, you're ready to go? How do you find patients? Okay, that's where the registries can really help us. How can you find the appropriate patients, right? Design of the trial. There you have to start thinking about all the foregoing questions I just raised about sources of variability. The reason we do trials is we can't just give a drug to patients unless it's like penicillin and pneumococcal pneumonia or something like that, and infer causal benefit. We can't do that because the disease, of course, is variable. So human variability is really the problem we're trying to deal with in doing trials. Now, usually we deal with variability by randomization. But again, we run into the problem. We have usually a variable rare disease, and we can't put many people in the trial because it's a rare disease, right? And so randomization may fail to deal with, under those circumstances, it may fail to deal with variability. So we need to understand all of this, both how many patients could be enrolled in the trial and what amount of variability we might expect on the outcome measures in advance before we start this trial. So, but because of the problem that we can only have very small trials, we need to do other things, if at all possible, to control variability and understand the impact of the treatment, so we can be convinced that the treatment works or not. We can categorize the patients. For example, we can group them by genetic mutations or other genotypic information, or by aspects, and that will enable us potentially to reduce variability and improve the plausibility of the design. Or we can characterize them, and this initiative is partly about characterization, disease characterization, right? What aspects of phenotype or disease course help predict the subsequent course or subsequent decline or what have you. If we have detailed natural history data on the impact of genotype or phenotype, for example, on disease progression, it will really help us in our confidence of designing a trial, how big the trial has to be, how much we have to match on these characteristics, and so on. So in sum, without having this information in hand, we're really wandering in the dark when we test an intervention. And the more of this information is available and the more clarity there is, the more likely it is that we can successfully test interventions and we can find the successful interventions. And we won't have our arguments or lack of understanding about whether an intervention is really doing something. And so to go back to what I said at the very beginning, people with rare diseases need treatments. We need to do what we can to make development of those treatments as efficient and effective as possible and as fast as when we hear from the patients as possible. And the way to do that is to have data. All this data I've been talking about the disease. So it can all be brought to bear on how we test that intervention. And the Rare Disease Cures Accelerator Data and Analytics Platform is a vehicle that can deliver that data to the developers and the community so that we can design trials with confidence that that test of whether they work or not and how safe they are will actually be informative, and we can draw conclusions from it. So there is really, in our current healthcare system and so forth, there is really no other way, I think, to accomplish this. This is the best we can do. And if we can pull this off, it will really, it truly will accelerate cures of rare diseases. So this is a long game, though. It's not something we are going to get done overnight, but I think in a year or two, we will start to see return on this investment. We will start to make progress in understanding first, and then developing treatments for rare diseases and improving the health of our population, which is what we want to do. So thank you very much for listening. And now I'd like to introduce Teresa Mullin. Teresa is the Associate Director at the Center for Drugs for Strategic Initiatives, and she brings about new initiatives, and if this isn't a strategic initiative, I don't know what is. So we're really lucky to have Teresa behind this. Teresa.
Thank you, Janet. So there's my first slide. So my job is to, well, good morning, everyone, first of all. It's my job to tell you about this construct that we've been talking about, the Rare Disease Cures Accelerator. What is it about, and what are its core components? And so first I'm going to, yes, it works, begin with trying to give some motivation for this and the context. And so the current model, and some of what I'm going to say in this talk, and I'm going to go through some of it quickly because it echoes some of the things Dr. Woodcock's talked about. And I'd say that's because when Dr. Woodcock talks about things, I try to pay attention. So take notes when she talks, and that's, so you're going to see an echo of what she said earlier. But the current model, this is motivated by the fact that this current model that we have is just not moving fast enough. What we hear from our reviewers is that they're seeing, one investigator's working alone in a lab. You've got families having to travel all over the country or the world to participate in trials or to get treatment or even to be diagnosed. One pharmaceutical startup with an idea is trying to move forward, and all of these individuals are very motivated and have a type of expertise. Each of them has expertise, but we're not getting enough synergy from this. And so they all come to FDA, or we may see all of them, and we're trying to piece it together, but what it presents is a very fragmentary picture. And also there are a lot of knowledge gaps that occur there. There's a lot of experience gaps. There's a lot of often a missing understanding of the regulatory requirements or what's needed for regulatory decision making. And so although we're getting a number of applications now, and it's really good, what our reviewers are also finding is that the quality of these applications, sometimes the quality of the natural history studies or the registries or the other information or the tests are not really validated tests. So what they're finding is the quality of what's being submitted impairs really good regulatory decision making, or even a certainty or enough assurance that this drug actually works. The evidence isn't where we want it to be, and really we're not serving patients very well by going to all this trouble, having people put all this effort into it, and at the end of the day, we still don't have an application that provides good evidence. So this is largely about also trying to pull these pieces together, standardize, and reduce any duplication of effort, take advantage of what synergies can be there. And so this is going to look really familiar if you're listening to what Dr. Woodcock talked about. I'm just going to say a little bit more about these three areas, not the only areas of critical activity, but three really critical areas of activity: characterization of the disease, getting patient perspectives on their disease, and clinical study, what's needed to put together a clinical study and run a trial, or run the studies that are going to be done in order to gather evidence about clinical benefit and the risks that are identified, and the challenges that are even amplified there for rare diseases. And so I'm going to just walk through real quickly, and this is truly a recap of what you just heard, so I won't spend too much time on it. But so the researchers and people who are investigating a disease have to really try to answer these questions. And for rare diseases, the challenges there are really compounded. So, what's known about the disease? Are there well-defined lab tests to diagnose the disease? In some case, a test may be used that's not validated, and the evidence that's collected is of whether there's any movement or improvement is questionable because the test isn't validated. The tests aren't standardized. Is the biomarker really been qualified for some purpose, for the purpose for which it is used in a study? So what do we know about the quality of the evidence? And what do we know about the natural history study and the causes of the disease? And all of these are very challenging questions, and if you've got maybe one investigator or two around the world and only a handful of people, the challenge of trying to answer the questions is that much worse. I hope that's readable. And then again, on the patient's perspective, assuming again that they have a diagnosis, that they are correctly diagnosed for their disease, what impacts matter the most to them? And could those be addressed by medical treatment? What would constitute meaningful change in those impacts for patients? And again, what would be your measurement strategy? What are people using today to treat the condition, if anything, and how is that working for them, and how burdensome is that? So those are questions that are important to answer also as you're thinking about measurement and moving into planning for trials. And with trials, you've got all these challenges too for rare diseases. Is there even an investigational drug available that can be administered? Has the basic safety testing been done, animal safety studies done on this investigational drug before taking it into humans? What kind of trial design is possible based on the information that you have, the number of subjects and so on? What is the study protocol, and how well designed is that? Is that going to be acceptable to patients? Is it tolerable? Can they get there? I mean, challenges of people being all over the world with the condition and the locations of the studies. And then IRB approval, submitting that IND to FDA. How are you going to enroll patients? And then what's the plan for collecting information from patients and so on? So just the usual kinds of questions, but made that much more challenging. We received I'm just going to go right into this slide. We see all these challenges, and I think there's a little bit of hand-wringing that goes on as you try to keep up with the individual and somewhat fragmented efforts. Congress gave FDA a special opportunity this past year, which we are now trying to take advantage of, and this meeting is part of this. Congress appropriated to the Center for Drugs $10 million for investment and innovation for rare diseases in this fiscal year 2019. And it provided us with a unique opportunity to try to put together this construct of a rare disease accelerator to try to start doing something systematic about these challenges that we see. And that's where this effort is originated. And so those three components of characterization of the disease, trying to understand the patient's perspective to try to figure out how to pull that into both measurement in the trial and also the design of the trial and how you run it, and perhaps even post-market what you continue to do to monitor patient status with the condition with treatment. So I'm going to say a little bit about these three components. And the data analytics platform piece that we'll be talking about today is foundational to everything else. It really will provide information useful to all three. So we need this centralized infrastructure to really have a better characterization of the natural history of a disease and rare disease development to support that, and to really pay attention to data standards. Now here's a case where we want to pull together different kinds of expertise, their data and their expertise. So we need people who understand analytics and developing structured data and cleaning data to have it be structured so that it's of good reliability. We know what it means, we know what the definitions are, along with pulling together people who have expertise in the disease and living with the disease condition and other components that we are still to be further fleshed out in terms of our work on this. And so we need this standardized approach to natural history studies in a common platform that can be accessed and used by multiple different diseases, multiple patients and investigators. That's kind of the concept. So this would begin with a platform that has a disease-neutral kind of background data, and then and a framework for that. And then on top of that, you would layer disease-specific needs that could be then standardized further as well. And all of this allows it to plug in other information as it becomes available. I think Joseph said earlier, if you have data related to this, they have expertise in cleaning and standardizing information to make it accessible to people who are working in development in that disease area. So that's the construct there, to try to take advantage of and not have people do multiple duplicative things in different settings and not able to get as far with their work. Another piece of this is the standard core clinical outcomes sets. So let's just say that developing a clinical outcome assessment set of measures that's fit for purpose, fit for regulatory decision making, can be lengthy and really resource intensive, and you got to have the methodological expertise as well to put it together, as well as the resources to run the studies and gather the patient input and so on. So CEDAR right now is piloting a grant program, where we are giving out cooperative agreement grants to highly qualified investigators who are pursuing development of standard core sets. And the grant announcement was for different disease areas We're awarding 3 of these. We've announced the awards last week, and 2 of the 3 will have good applicability to rare diseases. And so those are measures of pain and for infants and young children, and the other one is for measures of physical function for a variety of conditions for chronic disease, including rare diseases. So those are exciting near-term opportunities that we're pursuing. But the general idea here is that you develop a standard core set that has endpoints that have the minimum list of impacts that matter most to patients and are actually going to be able to measure a change as a result of therapy. So demonstrating change in clinical trials, and that we'd make that set available because FDA is paying for it. A condition of our grants is that that will become publicly available. So these are measures that people will not, we don't anticipate they're going to be costly to license in and use, as is the case with a lot of privately developed measures. So that's another important part of this platform. And the third piece, which is a little bit of a gleam in the eye right now because we don't have this one very far along, is the vision is a trial-ready network of investigators and clinical sites that would be for rare diseases and to really fast-track the moving forward and connecting up of patients with protocols and very highly qualified, trained investigators. And this platform would ensure some uniformity and training among investigators so that they are well-versed in what needs to be collected and done. Continuous collaboration and sharing of information will improve the expertise across the network as well, and just really kind of boost quality, and that the quality of the data can be improved by standardization, which is also possible by having that kind of network approach. And at the end of the day, this is going to produce very high-quality information that's going to help a lot of decision makers and do that much more to fast-track the development of rare disease drugs. And so with that, this is my last slide. And we're really looking forward to today and the launch of this Rare Disease Cures Accelerator. The data analytics platform really, as I said, provides an integral component and in fact supports the analytics that are possible here are going to support the other 2 components as well as the characterization, because it's going to really help identify, in fact, what are those clinical outcome measures that would be meaningful and measurable. And it's going to help inform clinical trial design and approaches there as well. So today's launch really is an exciting milestone for us, and we're really looking forward to what we have to hear from all the investigators, these patient groups, and others who are here today. So thanks very much.
So thank you for our morning presenters. We appreciate all that information. We're going to take a break and reconvene at 9:50. So please feel free to refill on coffee and food. Thank you.